I'm sorry because I just edited it, okay, with the PDF, but this came out with this, but it's okay. Right? This is the references that I use. You can have the other references as well. Get the later version. I put this because this is the only references that we have in KAIS. Yeah. So what is the animal tissue culture? Yeah. So basically, it's a technique to process of the keeping tissue alive and growing it in the culture media. Okay, you just imagine that you excise a chunk of tissue and then you try to mince it, make it into the individual cell, and then you grow it in artificial media. And the most important thing is you can make this cell alive and the cell can proliferate. Okay, the cell can proliferate. This is very this is very important. The cell can survive and continue to grow and be able to replicate. This is the ultimate goal for animal tissue culture. Okay, and not being contaminated, of course. And the growth of this tissue of living organism outside of the body of the suitable culture media, containing a mixture of the nutrients either in the solid or liquid form. You got to remember that when you excise the tissue from the animals, these cells, they are actually losing the immunity, so they are prone to be contaminated, but they are not being protected by the animal itself, the immunity by the animal itself, so it will lose the immunity and they are very fragile. So when you culture them, they can be easily contaminated by other microbes. So you need to make sure that you provide a conducive environment for the cell to grow and be able to survive and be able to replicate. Okay, and that's the basic of the animal tissue culture. Right, and this is a history. I'm not going to talk about this. You can just read it on. Yeah. So what are the advantages of tissue culture? First is the control environment. It's just in this environment, every single parameters that we employ can be controlled precisely. For example, our physiochemical environment, such as our pH, the temperature, the osmotic threshold, can be controlled precisely because we can monitor them. Okay, we can monitor this, and as well as the physiological environment, such as our hormone, the nutrients concentration can be kept constant, so you can monitor them. You can supply these the new hormone and nutrients at a constant level. Okay, so this is very important because the nutrients is a food for the cells to grow. If the cells is out of the nutrients, they do not have nutrients, so the cell may die. Because they are due to starvation, or the hormones are running out, so the cell might not be able to grow. So it's very important to make sure that the cells actually are being provided with the constant supply of the hormone and also the nutrients. And apart from that, the characterizations and homogeneity of the sample. So what you have is when you have this tissue sample, they are invariably heterogeneous. When you excise it, this cell may contain of different types of the tissues, but you need to subculture them for several times or to do passaging, so that a few times later after you subculture, then you have your pure cell line. That's the ultimate goal, yeah, of tissue culture. You don't want your tissue culture to have a mixture of the cell, because that is meaningless. So it is very important that at the end, when you isolate the cell, you want to have a pure cell line at the end. Yeah, and it's really easier for you to characterize the cell line later. Okay, if you have the pure cell line, but if you have the mixture of the cell line, is the data that you get will be totally wrong and it's not precise anymore. And in terms of the economy, scales and mechanizations, the culture may be exposed directly to the reagents as a lower and also defined concentration with a direct access to the cells and less reagent required. In terms of this, it could be a bit economical. And at the same time, if you do carry out the tissue culture, the legal and moral ethical issue can be avoided because we are not using the whole animal as an experimentation, but a bit of the tissue. So you can avoid this, avoid of the legal, moral, and also the ethical issue. Yeah. And also, you can have this in vivo modeling by provisions of the techniques that allow the delivery of specific experiments compound to be regulated in the concentration and metabolic state. So you can trace these cells, and then you can do a modeling, in vivo modeling, to look at the performance of the cell, right, during when you culture them. Right? Of course, you have the advantage, you have the disadvantage as well. What are the disadvantages of the animal tissue culture, which is involved, is the expertise needed. The expertise is very limited and it's very expensive also. And set up the facility for animal tissue culture could be very expensive. You go to the big company, for example, if you have been vaccinated with the COVID-19 vaccines, all these vaccines are being cultured, okay, for the weakened virus, okay, and you need to grow these viruses, and they employ the tissue culture techniques. So if you want to make sure that you have the state-of-the-art facility, of course you need to invest millions of dollars, and that is very expensive. Yeah, so this could be one of the disadvantages of tissue culture, and also the unstable nucleoid chromosomal constitution, whereby if you keep subculturing the cells, the genetics material could be very, very fragile or unstable. So this will lead to the unstable genetics material, will lead to the nucleoid chromosome constitution, right? Expertise we just talked about just now, and then the quantity also a major limitations for the cell culture, which because you put a lot of effort, but the amount, okay, the amount of the tissue that you get is very limited, right? It's very limited, and also the cell may go to de-differentiations, and the selection of the cell line could be also problematic as well, quite challenging, and also the origins of the cell, where the cell is originated from, also can be very challenging because you can sometimes trace where the cells has come from. Okay, and also instability, as I mentioned just now, the cell may be very unstable after you you culture them for several cycles, and especially the finite cell line, they are not be able to grow anymore. They will just go through the senescence of the genetics, and then no longer survive or be viable. Okay, so this is a major limitations for the animal and tissue culture. So far, okay?
Yeah, doctor.
Yeah. All right. So what are the types of the culture? We have the organ culture. For example, here we have the whole organ liver culture, liver culture. We have the primary explant here. For example, the primary explant culture of the pterygium's tissue, and also the culture of the congenital hamster ovary. And this is only the example that I can show. You. There are many others. Okay, many others of organ culture, primary explant, and also cell culture. Right. What is organ culture? Basically, it's the culture of the whole organs or the intake organs fragment with the intent of studying their continued functions or developments, and be able to accurately model the functions of an organs in various stage, and also the condition by the use of the actual in vitro organ itself. Okay, because sometimes you cannot use a whole or animal. For example, if we want to cure type 2 diabetes, we have type 1 diabetes, type 2 diabetes. Type 1 diabetes is come with genetics. Okay, it's born with, okay, where the person suffer from type 1 diabetes is the person that cannot produce the insulin. Okay, this is congenital, right? But for type 2 diabetes is the person that cannot produce enough insulin. They have the pancreas, but the pancreas cannot produce enough insulin, and that's lead to the type 2 diabetes. So if we wanted to culture the organ, for example, pancreas, and if we manage to culture the pancreas and we replace with the non-functional pancreas, and this is what we call a very good organ culture that we can replace. Yeah, we can replace it, and we can model the function. Yeah, because back in UK when I did my PhD, my next door lab, actually they are doing the pancreas culture, and they can be some of them they at the very beginning they can be very successful as well. So you just imagine there are millions of the patients with diabetic patients. So if you will be able to culture pancreas successfully. So you replace with the non-functional one, then the problem is solved. The type 2 diabetes can be solved at the root level. So they can keep producing the insulin, which is enough to convert the glucose into glycogen. All right. So primary explants refer to the small fragments of the tissue that adheres to the growth surface, whether it's spontaneously or aided by the mechanical means, such as the plasma clothes or the collagen that give rise to the outgrowth of the cells. Okay, and the primary culture refer to it that consists of the culture of the mixed complex organs or the tissue slice, which define a mixture of the cells and highly purified cell isolated directly from the organism. And this is the primary culture. Okay. But cell culture at general is referred to the culture of the cell that removed from the organs fragment prior or during cultivation. And the techniques of organ culture, we can employ few techniques such as the plasma clothes method, rough methods, agar gel method, and grid method. And we're going to talk about it in the coming lecture. All right, this is just an introduction, and this is the advantage of the organ culture. What is the advantage? First one is the explant that remain comparable to the in vivo organ, both in structure and function, and the developmental of the fatal organs is comparable. Information of the pattern of the growth, differentiation and development of the organs can be provided, and also can replace the whole animals in experimentations as a result from easier to interpret. Okay, however, the result from the organ culture are often not comparable to those from the whole animal study, because the whole animal is the whole system. Okay, and they are very complicated. But if you just culture the organ itself, the regulation, the physiological limitations is there. All right, so sometimes it cannot be comparable. So how are we going could culture the cell? How this cell culture can be obtained? Actually, in theory, it's very easy. So, for example, if you look at this, this is a fish. You can just excise a bit of the tissue from the fish. Then you mince it, or you chop it in finely, and after that you digest it with the proteolytic enzyme to isolate the cell into the single cell. And after that, you take the single cell, okay, and then you culture it in the culture dish. And this is what we call the primary culture. Yeah, it's very simple in theory, but when it comes to practical, there could be some challenges on this. All right. So this is also another illustration for you to obtain a cell culture. For example, here we have two different types of organ. One is a healthy organ. One is an organ that having tumor. So in organs, it's very easy. Also, you can isolate a cell using enzymatic isolation or mechanical action to isolate the cell. And after that, you culture the cell. When the cell become confluent, you keep sub-culturing them until you get the pure cell line, and that's you have the cell line. Right? That is the first condition. Another one, if you get this from the organ that having tumor, then you also can isolate the cell using the enzymatic method and also the mechanical method. But this cell, they consider as a transform because they are no longer the healthy one, but they are the tumor cell line. But you skip culturing them, you also can get the cell line. But this cell line is no longer considered as a normal cell line, but they are the transformed cell line. Yeah, the top one, this one is the finite cell line, but this one for the tumor cell line is what we call continuous cell line. Okay, I'll tell you the differences later between the finite cell line and also the continuous cell line. All right. So, what is adherent cell? Cells are encouraged, dependent, and propagate as a monolayer. Remember that the cell they need to encourage to depend on the nature of the cell itself. Some of the cells they do not need encourage. Okay. For example, like us, when we sit, we need something to support. Cell also the same. When they grow, they need something to support certain metrics to support them. Okay, that is what you call encouraged dependent, and need to be attached to the solid or semi-solid substrate for proliferation for them to grow. So they adhere to the culture of the soul with the use of an extracellular matrix. There are a few types, like polypropylenes. I'll discuss it in the coming lecture. So what are the cell types that belong to the adhesion cells, such as the fibroblast and also the epithelial cells, and the monolayer culture refer to one of the cell thickness, and majority of the continuous cell lines, they grow as the monolayer, right? This is the adherent cell. We also have the suspension cell, right? Suspension cell, that means they do not need to attach to any surface of the culture vessel. They are called anchorage independent or non-adherent cell, which can be grown floating in the cell culture, such as our hematopoietic stem cell, which derive from the blood, spleen, and also the bone marrow. And the tumor cell line can also be grown in the form of suspension. Okay, so these cell lines, they grow much faster and do not require the frequent replacement of the medium and can be easily maintained. Right? That's the difference between the suspension culture and also the adherence cell. Okay, suspension cell and adherence cell. Now, let's look at the finite cell line that I talked just now. Finite cell line basically is the cell line which go through a limited number of cell division having a limited lifespan that are known as finite cell line. Okay, and this cell line, they passage several times. Passage also meaning in the same meaning like subculture. You subculture them for some time, and then they are losing the ability to proliferate, which is the genetic determines event known as a senescence. They no longer be able to continue to survive or continue to replicate. Okay, and the cell line derived from the primary culture of the normal cell lines are the finite cell line, whereby the continuous cell line is a continuous cell line is referred to when the finite cell line undergo transformations and acquires the ability to divide infinitely. So such as our tumor cell line just now, the tumor cell lines is the continuous cell line. So such transformations or mutation can occur spontaneously or can be chemically or virally induced or from the establishments of the cell culture from the mammalian tissue. Mammalian tissue here refer to the tumor cell line or tumor cells. So the cell culture prepared this way can be subcultured and grown indefinitely as permanent cell line. Then they are immortal. You can continue like our tumor cell line. They can always multiply, replicate without non-stop. Okay, so this is the characteristic of the continuous cell line. So you must be able to differentiate between the finite cell line and also the continuous cell lines. Okay, so cell lines, they are less adherent, they are fast growing and less fastidious in their nutritional requirements. So they are quite easy to take care for continuous cell line compared to finite cell line, and able to grow up to the high numbers of their cell density and difference in the phenotypes from the origin tissue, and they can grow more in suspension and have the tendency to grow on top of each other in multi-layer on culture versus surfaces. But for finite cell line, this is quite not possible to do, right? So far, okay? Can you differentiate?
Doctor,
Yeah.
I have a question. So, continuous cell line is better than finite cell line?
Depend on the experiment, depend on the purpose.
Okay, thank you, Doctor.
Okay, we cannot say, oh, this one is better than no. It depends on the purpose.
Okay.
Depend on the purpose of the experiment. Yeah.
Okay, thank you, Doctor.
Yeah, because each cell line, they have their own advantage and disadvantage. So on the slide, you may look at, wow, the continuous cell lines is much better than the finite cell line. But when you really do it, it's not the case. Okay, so you need to be a bit flexible when you characterize or depend on your needs of your or the purpose of your experiment. Okay.
Okay, thank you, Doctor.
And also the influence of the environment on the culture is expressed for 4 routes. First is the nature of the substrate or phase on which the cell grown. So it could be the cell could be grown in solid, semi-solid, or in suspension, or physiochemical or physiological constitution of the media, or the constituent of the gas phase, or the incubation temperature. And these are the parameters environment you really need to take very good care in order to provide the conducive environment for the cell to grow. The cell can be very fragile. So where even the fluctuate in the temperature may greatly affect the growth of the cell line. Okay. So the provisions of the appropriate cell environments also include the substrate adhesion, the nutrients, the hormones, and also the substrate, which is also the fundamental to the expression of the specialized functions of the cell. The cell function may be losing. You may lose the cell function if all of these parameters are not really taken care of. All right. Now, let's look at the potency of the cell. So what does it mean by total potency, pluripotency, and multipotency? So total potency is the cell that have the total potential, where this type of cell that can be divided in or differentiated or specialized into any type of the cell. That's why you call the total potency. But this total potency cell, when they go through specialization or specification, they become pluripotent. That means this one, they cannot be divided or further be specialized into any type, but to certain type. Yeah. And when this pluripotent cell, they go through specialization and differentiation, then they become multipotent cell. Yeah. This cell, the multipotent cell, they have the restricted ability now, but they are more specific. Yeah. So if you learn mathematics, you can see the subset. You learn the set. Yeah. The multipotency is a subset of pluripotency, and pluripotency is a subset of the total potency. Yeah. So total potency here, this cell, if you call it total potency, that means the cells they have all the ability to be divided into or differentiated or the specialized specified into any type of the cell. Okay. So let's look at this illustration. So on top of this is a totipotent cell. Okay, so this totipotent cell, when they go through the specialization, they become pluripotent cell. And this pluripotent cell, when they go through the specialization, then they can become multipotent cell. And the multipotent cell here, for example, they can become a red blood cell, white blood cell, and platelet. And they belong to the same family. This totipotent cell, this group of totipotent cell can only be divided or further be differentiated and specialized into the red blood cell group. For example, if these cells go through another group, then they become the melanocyte, sensory neuron, and glia cell. So this another group of cell, which is totally different than the blood cell group. Okay, can you see that? Yeah. Down the graph, actually the cell become more specific. Yeah. Boleh faham nggak?
Boleh, doktor.
Boleh. Boleh.
Berapa? Saya reply text. Okay. Is that okay now? Right. So can you see the differences between the totipotent cell, pluripotent cell, and multipotent cell?
Yes, doktor.
Yes,
Yeah.
doctor.
So if you want to if you want to know more about this totipotent cell and pluripotent cell or multipotent cell, you can read an article from published in Bioassay by Robert Kelch in 2006. Robert Kelch is my PhD supervisor, a very smart professor from UC of Cambridge. Okay, so you can read on this paper, yeah, to be able to understand more about totipotency, pluripotency, and multipotency, right? And so I'm not going to talk about this. Like the pluripotency in the stem cell, for instance, the in vivo fertilized eggs, this egg, when this egg is fertilized, this is what we call the totipotent, because the cell can be, this fertilized egg, they can form into any types of the cell. But when the cell are going through the cleavage or division, okay, or division, then they are having different types of potential. For example, during the blastocyst, if you get the cell from the blastocyst, then you culture them, that cell become a pluripotent cell, and this pluripotent cell can be further be specialized or differentiated into the neural cells or cardiac muscle or blood cell. Depend on the route where the cell is being instructed to. Depend on the end phase of the cell. All right. I've talked about this also. So what is the de-differentiations? Basically, de-differentiations is the irreversible loss of the specialized properties that a cell would express in vivo, and especially or terminally, a differentiated cell revert to an earlier development stage, usually as a part of the regenerative processes. All right, you can read on. You can look in more details of what is the definitions for differentiations. All right, so you have assignment for this for this lecture. So I have posted it, I have amended it you can find the more extra information in Ellipse. So what you need to do is to discuss the applications of the animal tissue culture towards the future medical or technological advancement. And you need to form a group of 5 to 6. And then the deadline is on the 22nd of November. And this is between 3,500 words to 4,000 words. And you can discuss based on these topics, okay? The model system, the toxicity, the cancer research, virology, up to drug screening and development. Choose only one topic for this assignment, okay? Right?
Okay.
Okay. You can look at the instruction on Ellipse later. I have just uploaded the information on Ellipse. Okay. And so I think that's all for today. It's a bit short actually. All right. Any questions so far?
So far, okay, doctor.
So far, okay, huh? Everything clear? Yeah. Yeah. Go ahead. Okay, now, apa itu differentiation? Do you understand what is differentiation? Okay, de-differentiation adalah sebaliknya. Okay, dia sebalik pembezaan. Kita ada sel pembezaan sel kan? Okay, so de-differentiation ialah di sebalik pembezaan sel itu. Faham tak? Okay, contohnya sel itu dia mengalami pembezaan. Pembezaan kerana contohnya sel group type A ini. Bila dia mengalami sel pembezaan, dia akan menjadi grup mungkin sel type C, D, E, F, G. Betul tak? Ya, sel pembezaan kan. Tapi de-differentiation ni, dia sudah menjadi B, C, D, E, F, G. Tetapi dia de-differentiate menjadi B semula. Basically itu maksud dia lah. Okay. Right. Kalau tak ada soalan, siapa belum ambil kedatangan? Ada lagi tak yang belum ambil kedatangan?